Okay. Sorry, but I just need to stream my thesis. Yeah. Okay. So 6 of the top drugs highlighted here are produced using single platform, which is the Chinese hamster ovary cells. So the reason why people prefer Chinese hamster ovary cells is because when our target genes are transfected into the genome, into the cell line, it's very easy to integrate it into the genome, and then we can amplify. It's also very easy to amplify it, so we will get a lot of copy numbers for our target genes. And in this case, we can produce a lot of the drugs we want. But even though CHO cell is preferred by most of the companies, it still has some challenges for using the CHO cell line. So first challenge is the production. So we always want to produce more proteins using the cell line, but before we don't have much resource for CHO. So what people did is they tried to use different cell culture or try different chemical compounds to see if it improve the production or not. And it's very inefficient. And nowadays, it's almost hits a limit. So we need to come up new strategies to improve the production. And another limit is that we not only want the cells to produce antibodies, we also want the cells to produce high quality antibodies. So in that case, we need to we want to control the cells to produce a certain format of antibody. So in that case, we can get high quality antibodies. And to do that, we need to know more of what is happening inside the cells. And another, lastly, the big challenge is the purity of the products. So 80% of the money actually was spent in this step by the companies when they produce proteins. They need to purify the protein, and they also need to make sure the products doesn't have any contaminants, especially the viral particles I will talk about later. So in order to kind of either solve these challenges or kind of provide some improvements to kind of tackle these problems, I'm going to focus on different aspects of the central dogma. So this is a central dogma, so which means here we have genomics, and the genes are kind of transcribed into mRNA, and mRNA is translated into proteins. So my thesis focused on different aspects of this central dogma to either provide high quality resource for the community or show how I can apply the cutting edge technology to improve the performance of the cell line. So first I'm going to focus on the translation level. Yeah, so translation level, as I mentioned before the challenge, the cell does not only produce antibodies, it also produces many other proteins. So we have a question that what else are the cells are producing? Because the reason we care about this question is that to produce proteins, the cell needs to spend some resource and cost some energy to produce that. Then it comes to our next question. If we can find some distractions in the products, and if we can remove those distractions, our hypothesis probably can we improve the production or not? So these two questions, to solve these two questions, we decided to use RNA-seq and RiboSeq. RNA-seq measures the transcription level, and RiboSeq measures the translation level, which I will explain details later. And we didn't apply the proteomics here because proteomics is a little bit sparse, so it cannot measure all genes. So, and I think most people here are familiar with RNA-seq, but probably some people are not familiar with RiboSeq. So I'll explain how RiboSeq works here. So the RiboSeq, basically, you have a cell. And first, we use lyase to remove the cell membrane. So after you remove the cell membrane, the ribosome will still move along the mRNA to translate proteins. And at this time, we apply a drug. It's called cyclophosphamide to stop the ribosome from moving. And then we use another enzyme to degrade all the sequences that is not being protected by the ribosome. And then we left kind of 30 base pair length. We extract all those reads, and then we do the sequencing similar to RNA-seq to get high throughput sequencing to measure the translation level. And the experiment design is we chose two phases here, a log phase and a stationary phase. So these two phases are usually the cells produce more proteins, and it's being used. So in our case, is that day 3 is the log phase, and day 6 is the stationary phase. And we, for both phase, basically we apply both RNA-seq and RiboSeq, and to get the high throughput sequencing. I just briefly explain how we kind of pre-process the high throughput sequencing data. So first we kind of remove some contaminants RNAs, and then for the rest of the reads, we just map back to the genome and to quantify that. But to quantify the reads, there are some differences between RiboSeq and RNA-seq. So RNA-seq is straightforward; it just count how many reads map to the coding region. But for RiboSeq, it's a little bit different. So to understand how different it is, so let me explain how ribosome works in the cell. So this is the ribosome. It has EPA site, and it has the large unit and the small unit. So when the ribosome translates protein, the A site has an amino acid, and then it forms a bond, a peptide bond. After it forms a peptide bond, the big subunit moves 3 nucleotides forward, and then next the small subunit moves 3 nucleotides forward. And then in this case, the A site is empty, so the next amino acid can come in and form the new bond here. So this is kind of how the ribosome works. And then let's see the coverage for the RiboSeq. It's a little bit different from the RNA-seq, which RNA-seq is usually each location, the read is kind of similar, coverage is similar. But for RiboSeq, we can clearly see that all the reads map to only the coding region. And also you can see here the coverage follows a 3 nucleotide periodicity, which reflects what I talked about just now. The ribosome moves in the unit of 3 nucleotides. So in this case, we can get a very high resolution of which amino acid the ribosome is translating at that time point. It also helps us to define the translation frame, because you can see that usually the first frame is the most ribosome located. And so after we know the difference between that, then we are ready to do the quantification for each gene to calculate their transcription level and translation level. But before that, we also have a question, which is, since antibodies are not endogenous genes, they are transfected into the cells. So we have a question that, are those antibodies kind of have the similar patterns compared to the endogenous genes? So in order to answer that question, so what we did is we basically compared their read coverage situation patterns and see if they are similar or not. So this one shows that RiboSeq and the RNA-seq coverage. So we can see that RiboSeq maps only to the coding region, and if we zoom in to some certain location, we can see it has the 3 nucleotide periodicity coverage. And if we overlay all the reads in each frame, we can see that the first frame has the most read, which kind of makes sense. And let's see what about antibody. So the antibody here is the guide to see they have the similar patterns. So then in this case, we can kind of treat them as the same in the same protocol and to quantify the read. So this is so since we have RiboSeq, I also have RNA-seq. So we basically calculate we quantify them, and we do the scatter plot to see how they correlate. And this one shows how they correlate relatively well. And these are 3 genes: the heavy chain, light chain, and the NUR. It's a selection marker. And the benefit here of using RiboSeq, well, we can answer another question. So using RNA-seq, we can know that those antibodies are being highly transcribed. So with RiboSeq, we can answer another question: are those antibodies also being highly translated? Which means, which of these 2 cases can best describe the translation of antibody? Which means, for each single mRNA of antibody. Does each mRNA of antibody has more ribosome compared to the endogenous genes, or it has the similar amount of ribosome for each gene? So in order to answer that question, so what we did is we used the RiboSeq divided by the RNA-seq. So intuition between for this is that it measures the number of ribosome in each mRNA for each gene. And so we can see here the three genes are kind of similar to most of the endogenous genes, so which means it's this case. They are being highly transcribed, but in the translation level, they have the similar translation compared to all the other endogenous genes. And some people may ask, may have question that why, because most people, what we care about is the antibody, is the heavy chain and light chain. Why do I list neomycin here? The answer is in this slide. So this is a protein map. So protein map, basically, you can consider the total area of this shape. It reflects the total amount of ribosome in the cell. And each subunit, kind of like a small fraction, it represents how many ribosomes that the cell allocate to each gene to translate them. So kind of like a proportion of ribosome goes to translate each gene. So we are happy to see heavy chain and light chain are highly translated here. But the thing is, we also see this selection marker is also highly translated. Now this is for an exponential phase. So let's see what's the case for stationary phase. So the good thing for this protein map is that you can go multiple layers. So here I will just show a layer up, which means for each functional module of the cells, how much of the ribosome are used to translate those genes in the same kind of functions like that. And so heavy chain and light chain are still highly translated, but NEOR is also highly translated. So in this case, so we compare for these kind of functional modules, we compare day 3 and day 6. We see that here, the antibodies use less percentage of ribosomes in the stationary phase, but the NEOR is still use the same amount percentage of the ribosome. So remember the second question I asked in the beginning. Can we remove some distractions to boost the production? So in this case, the NEOR gene can serve as a perfect, could be a kind of potential perfect target as a distraction. So if we remove those NEOR, can we release the ribosomes? So those released ribosomes, if they can go to translate our antibody, then we can boost the production because it can occupy a large chunk of the ribosomes. So we gave this strategy to our experiment team in Denmark, and so what they did, they used siRNA to knock down the gene. This is a kind of proof they successfully knocked down the gene. So you can see that. So this is a Western blot. You can see it's correctly knocked down the gene, and this is the proof of knock down the gene in the transcription level. And so after we knocked the gene, then they did the culture the cell again, and they found that the cell density increased by 35%, and the titer increased by 18%. So this is kind of like we use measure the translation level and find the target, and then give strategy to the team, and then kind of improve the performance of the cell line. So basically, the summary is that RiboSeq, it can measures protein synthesis. The other methods kind of, because it's measured in the real time. And another thing is it has very high resolution. You can, with RiboSeq, you can know the exact amino acid that ribosome is translating at that time point. And another thing is it provides a new measurement of the cell line production, because we can come up with new strategies and to help them to improve the production. And next, I'm going to focus on the genomic side. Because the big challenge in our community so far is that the genome is very fragmented. So for the fragmented genome, most of the time for the quantification studies, sometimes it's okay because the annotation has allow some missing of the exons, and you can still get some quantifications. But if you want to do the other studies like CRISPR, or you want to study mutations, then you need to have very precise, specific sequence in that region. So in that case, we need to have much better genome. So let me first review the situation we locate right now before. So in 2013, we have 2 genomes for a hamster. These two, we can see that the scaffold number is pretty high. It's in the thousands of numbers of the scaffolds. And also the gap region is also very high here. So our collaborator in Germany, they kind of decide, because before they only use luminaries, so there is a lot of fragments and gap regions. So what they decided, they use the PacBio region, PacBio reads, and combine them with the previous assembly, and they generated a much better genome. It's more continuous, and it has less number of scaffolds. So this is the kind of statistics. It shows that it reduces the scaffold number by almost more than 20-fold, and the total gaps is also reduced, substantially reduced. So, what I focus on is that I compare the RefSeq with because this is what people most of the time they use. I compare this one with the new genome and to see how much the genome has been improved and what additional information we will get using the new genome. So first, let's look. I'll look at the gap region. So this plot is basically shows compare the gap regions between old genome and new genome. PSCR PSCR is a new genome, so we can compare them, and we see that you can barely see the gaps for the new genome here. This is reduced a lot of the gaps. So let's take a log phase and see how much it's improved. So if we take a log 10, and we can see that it's basically reduced the number of gaps in orders of magnitude. So it's a huge improvement. And then as I said, I care about what information are in those gap regions, and are those information important? So, but to answer that question first, we need to identify the gaps. So the strategy I used to identify the gaps is that I used a tool called MUMMER. Basically, it can do the genome scale alignment. So, and the strategy I did is I take advantage of the fragments on both sides of a gap, and I align them to the genome. And then I compare the gap length with this region to identify the sequence in the new genome and to say they were gaps before. And this is the kind of perfect case, but there are many other cases, like for example, for sometimes the gap in the PSCR is shorter, sometimes it's longer, and sometimes it's like this. So the gaps is too short, which means aligner will continues to align and generate one single fragment. So after we get this, then we find all the gaps. Then we are interested to see what information are there. So the way we find information, we care about the information is what genes are in the region, and also what mutations in those regions, because those are kind of affect a lot of studies if people use the genome. And the way we find the mutations and genes in those gap region is what we did is in the new genome. So we have several DNA-seq samples. So we basically, what we did is we did variant calling using those DNA-seq samples, and we identified mutations, and then we overlap the mutations and also annotations to identify any genes located in this region and also the mutations located in the region. This is the result we found. So most of the mutations we can see it locates in the non-coding region. Now it's a kind of a little bit relief because at least it says the old genome, at least it's not in a very poor quality. It still has some values to use the old genome, and we can see that the valuable thing here is that there are some mutations located in the coding region. And for the coding region, you can see most of them are SNPs, and some of them are insertion deletion. It makes sense because I use GTK to call the variants, and GTK tends to call more SNPs than the insertion deletion. And then after we find this, then our question is that is there any important kind of mutations in those ones that kind of really affect some studies? Here's an example. So our collaborator Jeff Esco, professor in medicine department, so he basically developed a CHO cell line. That cell line is resistant to some virus. And the reason why the cell line is resistant to the virus is because there's a mutation here in the gene called xyloside transferase. And that mutation is a loss of function mutation, which means the result of this is that the cell was not able to synthesize the heparan sulfate. The heparan sulfate is a receptor for some virus, so which is why the cell is resistant to the cell line. So he kind of find this very specific cell line, and kind of it's very important. And later, as the NGS technology develops, he get the sequence, and then he wants to test if the mutation, if we were able to identify the mutation using the NGS technology. So he did a whole genome sequencing and collaborate with us, want us to find if the mutation is there or not. But surprisingly, we didn't find a mutation. So what happened? And so we kind of find a lot and discuss with a lot of my PIC if we should tell him the results or not. But yeah, but but after we did some debugging and kind of look into the details, actually, it's none of us are wrong because this is the O gene, and we find that the first exon in the old genome, the beginning part of that region is in the gap region. And so since we have the new genome, then we double check, and we make sure that the mutation actually locates in the gap region. That's why we were not able to find it. So, and to further verify that, we also include other DNA-seq samples we have, and we did variant calling, and we compare with the specific sample, so we can see that all the reads in that specific cell line, the read supports the mutation, and for the other cell lines, they support the reference. So in this example basically shows that if you use the old genome, sometimes you could probably miss some important mutation, especially if you're doing discovery studies like that. So the summary for this one is the new genome kind of filled most of the gaps, and also, as I said, there's some coding region and mutation we missed if we use the old genome. So after we have a better genome, the next step is that very important is that we need to have a high quality annotation, because that would affect all of the downstream analysis people are doing. Here's an example why it's important. Our collaborator in Denmark, CRISPR is very hot these days, so they were trying to use CRISPR to knock down some glycosylation gene to see how those genes correlate with some phenotypes. So they tried to use CRISPR to knock down those genes, and they failed. So the sequence they use is that they get the gene sequence and annotation from NCBI version, and then they so because they want to target the transcript, the promoter region, so they just used the annotation from NCBI and extract the sequence and then target it, but it failed. So they come to me and ask me to kind of check what's wrong with annotation. Is there something wrong or with the sequence? So since at that time we already have the new genome, but it's not public yet, so I was able to see what's the problem. So I aligned the RefSeq to the genome. This is the new genome. And since we also have RNA-seq samples, so I also align RNA-seq samples to the genome. So here's the results. So the red bar here are the alignment results of the RefSeq version of the gene. And the gray bar here are the alignment results of the RNA-seq samples. So we can see that the last two, they match each other, but the first exon, they're different. So I think probably that may be the reason why they fail. So I provide this sequence to them, and they get upstream of this region, and they succeeded to knock down the gene. So this is kind of proves that if you use the wrong annotation, then you will have a lot of problem in the downstream analysis. So with this, then we use a strategy. We collaborate with Dr. Vinay Bafna labs to use proteogenomics method to basically generate a high quality genome annotation. So the project is, so first we have the proteogenomics basically it's combining RNA-seq and the proteomics to give a very high quality of defining of the coding region in the cell, in the genome. So first we have genome, and then the next step, since we have the spectrum, so we need to map our spectrum to some protein database. So we need to generate some kind of protein database to map to. So first I generate the draft annotation, which I'll talk about later how I generated. And those ones, they have some known database. We call it known database because we use some known protein or resources to map. And then we also generated some novel peptides that is not in the draft annotation. So for example, RNA-seq samples. So we have RNA-seq samples from multiple tissues of hamster. So we want to cover as many genes as possible. And so in that case, it can provide you additional information like new novel splice sites or mutations like that. And for RiboSeq, as I mentioned, you remember the coverage of the RiboSeq. So it can help you refine the coding region boundary, and also help you to define the translation frame using the RiboSeq. And the last protein database we used is the 6-frame protein, which means you have 2 strands, and for each strand you have 3 frames of the you have three frames, so you can translate the proteins and generate a database. So for those database, after we generate those database, then we can align the spectra, mass spectra we have gathered. I think it's 12 million spectra, and we map them to the database we generated, and then we get peptides. And for those peptides, we kind of map back to the genome, and then we identify the known protein, or if they are novel proteins like that, or this one is another important retroviral proteins, which I'll discuss later. So first, let's discuss how I generate the draft genome annotation. So as I said, we have multiple RNA-seq samples from multiple tissues of hamster. So for each sample, we mapped the reads to the genome, and then we did a transcripts assembly to generate the transcripts. And then we also have high quality RefSeq. So we mapped our high quality RefSeq to the genome. And then we use a tool called PASA to merge these transcripts to generate a union set of high quality transcripts. And then we use a transcript coder to predict protein sequence in those transcripts. And then after we get those proteins, we map to the RefSeq and UniProt and see to kind of help define the function of the proteins we predicted. And so since we get the draft protein, and we want to see how much the draft protein has improved compared to the old RefSeq ones. So in this case, there are we aligned the RefSeq to the draft version of the protein sequence. So there are several cases of mapping between them. And here, high quality mapping, what I define is similar to, I think, the Ensembl and NCBI when they do the mapping. So the high quality mapping basically means 90%, the percentage of mapping is over 90%, and the map length of each protein is over 80% of the total length of each protein. This is what I define as high quality mapping, but there's some difference between them. So another case is that it's called RefSeq perfect, which means RefSeq can map perfectly to the draft protein, But since we improve our annotation, so the draft annotation is longer. So it basically it generates longer proteins compared to RefSeq. And another case is that it's similar to the RefSeq perfect, but in this case, it's not perfect. It just has high quality mapping. But draft annotation, it's not kind of a high quality mapping compared when it maps to the RefSeq. And another case that it's got a draft high quality mapping, which means the draft protein, it maps well to the RefSeq, but RefSeq probably longer than that, and the RefSeq the quality is not good. And the rest of them are considered as low quality mapping. So for each of them, since we identify the peptides, so we map our peptides to both RefSeq and draft, and we compare. So if there are additional peptides, especially if they're unique, maps to support the draft ones, then we consider that draft protein has higher confidence compared to the RefSeq ones. So this is the results. Basically shows. So the left here shows that for each of the protein in the draft protein here, it has unique peptides support them, which means they are probably true compared to the RefSeq ones. And the right side is that it has less quality because it doesn't have unique peptide support them. All the peptides support them, they also map to other positions. So in that case, we are not that confident to say that the draft protein is better than the RefSeq here. So after we compare the known proteins, then let's talk about the novel proteins we identify using the pipeline. So this is the results. Basically, you can see that most of the events are novel splice and the SNPs identified using the pipeline. And this is going to be statistics, see how many of them are kind of supported by the peptides. And we're going to publish this. So basically, this would be the resource. So when people do some studies, they have some mutations or they have any kind of novel translation events, they can look this up, see if this has been identified or verified or not. Okay, so here I'm just going to show one example how I improved annotation. So for example, here you have mRNA and it has an annotated protein. But if you can predict another protein and it has peptide support, then in this case we need to kind of split this one because these are actually two proteins. And another case is that it's annotated as non-coding RNA, but you have predicted protein and you also have a peptide supported. In this case, it should be annotated as coding RNA. So this is kind of how I improved annotation. And after we improve annotation, then we want to see because our proteomics data, we have 3 cell lines per CHO. And this figure basically shows how people develop the cell lines as time goes. It's kind of evolution. So since the peptides can help us to identify the mutation, so we're interested to see. So we have these 3 cell lines. So we're interested to see how much difference regarding the mutation are these 3 cell lines have. So in this case, we can see that for all the mutations we identified for each of the cell line, they share most of the mutation, which means they diverge from the parental cell line, but they still share most of the kind of mutations compared to the reference. And also, but at the same time, they also have their own mutations. So this figure basically tells us that those 3 cell lines are not that different. They have some kind of certain features very common, but probably some mutations that cause them be different as they evolve. And since we have proteomics, let's go back to the as I mentioned, the last challenge of this one is that FDA requires the company to check if their products has virus or not. So this is a very serious question because they want to make sure the drug is safe for people to take. So since we already have the proteomics, so we can use the proteomics data to verify if in the products are there any kind of translated viral particles. If there is, then we probably can come up with some strategies to remove those retrovirus. So it has already been shown that CHO cells can produce the active full length of the retrovirus before. So, so in this case, we want to use the proteomics to verify if. But since this the transcription level has already been verified, so what we did, we can take advantage of our proteomics data to try to verify in the translation level, and then we can probably come up with a solution to remove that. And there's one good example of kind of provide positive feedback of the solution. So basically, in 2015, there's a George Church lab. What they did is that they successfully used CRISPR to knock down 60 copies of retrovirus in the porcine cell line, and 2 years later, they kind of generate a real pig that doesn't have all the retrovirus. So their final goal basically is to use these pig organ to transplant to human. So that's why they need to remove the retrovirus to kind of remove the risk for the transplantation. So with that, then it kind of this example basically gives us positive feedback. So which means if we identify all the retrovirus in the CHO genome, we probably can use this method to remove those retrovirus, and that would be save a lot of money for the companies to kind of remove those retrovirus. So, but before to remove those one, first we need to identify all the retrovirus in CHO genome. So this is the kind of basic structure of retrovirus. You can see that. So it has long terminal repeats here. That's the structure. And there are 3 main genes in the retrovirus. It's called GAG. It's encoded capsid protein. And the PO here, it encodes the reverse transcription, some other enzymes. And AMP here, it encodes the envelope protein. So these are the main 3 proteins that we care about. And if we can identify all the loci that are retrovirus, then we can target them. So the strategy to find those retrovirus is straightforward. So in our draft annotation, we already have some annotation. So in that case, then we just need to see if there are any peptide support. And if they are not in the annotation, then what we did, we download all the retrovirus protein sequence from NCBI, and then we align them back to the CHO genome and see how much they align to. And if they align, then we try to identify if their peptide support them. Because this is a result of the numbers of the viral proteins we find using the peptide support. And another thing is that what we care about is, because usually if your protein is very long and you only have one peptide supported, we probably are not confident enough to say if this protein is being produced. So usually we are confident to say it's produced when there are many numbers of peptides supported, and also the total protein body is being very well covered. Then we can say, well, this probably has higher probability of being produced. So this figure basically shows that each dot here represent a protein loci, and the x-axis is the peptide number support at retroviral protein, and the y-axis is the proportion of the protein is being covered. So which basically means for most of the proteins we identified, they are in this region, which means we're probably not confident to say they are highly kind of produced. But there are a few cases here they have high coverage, and also they have a lot of peptides support them. Those ones probably mean they have higher quality. So this is not the end of the project, because right now we are collaborating with another company. The company is because here we use the kind of a proteomics target all the things. So our next step is that we apply the PacBio IsoSeq to get to kind of target the virus directly and to get its whole sequence of those ones. And also we generate proteomics for those retroviral particles to see if we further verify if those retrovirus are produced or not. So this is kind of a summary of the annotation project. Basically, high quality of the genome annotation kind of enables accurate genome engineering. As I mentioned in the beginning of this project, before I kind of collaborated with the Denmark team to provide a corrected sequence. And proteomics basically can helps to improve the annotation, and it also helps to identify the proteins we want to find. So this is kind of summary of my thesis. Basically, these are the challenges CHO cells are facing when people do the study. And so my thesis basically is a central dogma. So my thesis kind of focus on each aspect of the central dogma to either provide high quality resources for people to use in the downstream analysis, or kind of provide them an example to show how to use some cutting edge technologies to improve the performance of the cell line. And this is not the end, because some future directions, we can see that we have a much better genome right now, but it's not the end, especially if we compare with human genome or mouse genome. So the CHO genome, the hamster genome right now is kind of still have kind of some steps to go, and especially in the chromosome level. So to solve this, since we use PacBio read, PacBio read is long, but it's not long enough to cover all the repeats. So in that case, we probably can, in the future, we can use other technologies like optical mapping or Hi-C like that to kind of further tackle the structure of the genome. So in that case, we can get a probably better genome, because I think that we can see that there are still some gaps in our new genome, and probably there are some additional important information in those ones, and we're looking forward to improve that one. So another good looking is that since we have a much better genome, so I think it will help a lot for the downstream analysis, especially when people right now these days CRISPR is very hot, so everyone is using that. So this one is definitely the high quality genome and high quality genome annotation will definitely helps a lot of people in our community to do a lot of analysis, especially, for example, not only confined to the single gene manipulation, they can also do the kind of CRISPR screen to build the genotype to phenotype correlation like that. So there's a lot of applications for those ones. And with that, I'm gonna to close here, and I would like to thank my committees, especially my PI, Nathan Lewis here. He helped me a lot during my PhD, and not only gave me advice on the research, also helped me other career development and also any other stuff. And I also would like to thank Vinay Bhavna, Professor. We collaborate with them on the proteogenomics project. Basically, they provide us the pipeline to analysis. And I would also like to thank Professor Hannah Carter, Professor Kun Zhang, Professor Xiaohua Huang to give me some good advice for my thesis. And I would also like to thank my lab collaborators. So made a lot of friends, and especially for the food adventure, it's really a good time there. And also thanks to other UpLab collaborators. Without them, I cannot finish my project, especially for the experimental part. Yeah, thank you so much. I think that's the end for my thesis. I'm open to questions. Mm-hmm.
So for these electrolysis, you have to take the database from like non-managing electrolysis.
Yeah.
did you not find any transcript evidence for them? Only proteomic, or did you have both transcript and
full proteomics for the part of the
viral identification? Why was
yeah.
not found in just in genomics? Transcript derived.
Yeah, but for transcripts, we kind of find some of them, but transcripts does not necessarily mean the protein is being produced. In that case, yeah. So that's why we kind of want to focus on the protein level too, since we already have the proteomics data. So we have kind of this advantage to apply this to see if there are real protein particles have been produced. Yeah. Okay.
Why did you need the additional retroviral genomic sequence? Why did you not find them in your original proteogenomics?
Yeah, I think that's the limitation of the draft annotation thing, because in the draft annotation I created, basically I assembled the RNA-seq, and that includes the mapping. And for the mapping, sometimes it's short read, and for short read, it's as we know, the retrovirus is very repetitive. So the mapping is actually, we probably have some problems for the mapping. So in that case, if we, for the draft annotation, they may not recover all of them. So that's why we use the additional resource to do that. Yeah.
So I'm trying to get an idea for like how much you've done in this in your process. I think it's a lot. But I mean, if you've taken us, 2 orders of magnitude closer to a high quality genome, 12 orders of magnitude, how many orders of magnitude away are we from a high quality genome for Chinese hamster ovaries?
Yeah.
Where were we and where are we now?
Yeah, I think regarding that depends on what kind of analysis research you are doing. So regarding the kind of genome quantification, I think we are very close to kind of perfect, because most of the time the genome annotation, it's not limited by some of the genome rearrangement like that. So, but if we were doing the kind of those kind of analysis like the genome rearrangement, or we want to do study in the whole chromosome level, we still have a long way to go, I have to say. But regarding those kind of quantification or some mutations in the genes, that would be already good enough for the genome. Yeah, I have to say. Any questions?
Another question.
Yeah.
So, Chinese family, right now, what percentage do you have a number?
What percentage of the genome has been completed? Here, let me see the statistic here. It's because here basically shows that so the Chinese hamster genome is similar to. So Chinese hamster genome is the genome size is similar to Chinese hamster genome size is similar to human size, but it's a little bit less. So people's estimation is that it's around kind of 2.4 G base pair, and we kind of cover, I think, probably over 99%. I think for about 95% of the genome recovered. So there's still kind of probably 5% of the genome still need to be recovered, but it's obviously right now it's in a very high quality. It's just a matter of we need to further merge the fragmented scaffolds into longer ones. Yeah, but for sequence level, we already covered most of the sequence.
Did it include centromere, telomere, or the?
Yeah, for those one, I think we are still don't have that information. We only have, so for those scaffolds right now, we only have what chromosome does that kind of belongs to. But for those structure ones, we still haven't done that yet. So we still need to further improve the genome annotation, genome assembly. Yeah.
no more questions, and everyone on the committee, step up, so we'll have the closed session with Sean.
Oh, sorry. Yeah, sure. What?